Okay, so welcome to this next video in which we are discussing the mammalian circadian timing system. Okay, so we're currently in the process of discussing how these photosensitive retinal ganglion cells are going to be able to make sure that the suprachiasmatic nuclei neurons are oscillating in tune with the light-dark cycles. Okay, and we've seen that what's going to happen is when it is light, the photoreceptive retinal ganglion cells are going to be firing and they're going to be releasing glutamate onto the neurons of the suprachiasmatic nuclei. Okay, and this is going to trigger a calcium signal within these neurons of the suprachiasmatic nuclei, which will trigger the activation of calcium calmodulin dependent kinase 2 enzymes, which are now going to go into the nucleus and alter transcription. Okay, and they're going to increase the transcription of period and cryptochrome genes, and this is going to then reset our internal circadian oscillators so that they're in tune with the light-dark cycle. Okay, right. So we want to see how is the calcium calmodulin dependent kinase 2 going to activate the transcription of period and cryptochrome. Well, it's going to work through proteins known as Reb proteins. Okay, so I'd just like to take a little deviation off to discuss Reb proteins. So Reb stands for cyclic AMP, response element. So the C is for cyclic AMP, the R is for response, the E is then for element, and then the B is for binding protein. Whoops, get rid of that. Element binding protein. Okay, right. And these proteins, because there isn't just one Reb protein, these proteins are transcription factors which are going to change the transcription levels of certain genes. Okay, so first I want to discuss the different types of Reb protein. Okay, so there are three main Reb proteins. There are more than three, but the other ones are very niche. Okay, so the three main Reb proteins are Reb alpha, Reb beta, okay, and then finally also Reb delta. So these are the main forms. Now, they are not actually produced by different genes. They're produced by the same gene, but they are different splice variants of that gene. So let me just explain the concept of splice variants. Okay, right. So, let's say these two parallel lines here represent the two strands of DNA. Okay, and let's say that this portion of the double-stranded DNA, which I am now boxing, let's say that this is going to be the gene for Reb. Okay, so this portion I'm now highlighting in turquoise, this is going to represent the Reb gene. Okay, now, those two strands of DNA, only one of them is going to actually be used by the RNA polymerase enzyme to actually produce a piece of mRNA from. The one that is used by RNA polymerase 2 to make a piece of mRNA from is called the coding strand. Okay, so let's say that this is the coding strand. We've been through this already. Okay, but I'll just remind you. So this is the coding strand, and then the other strand that's not used by RNA polymerase 2 is then the non-coding strand. Okay, so in blue here, this is the non-coding strand. What then happens is RNA polymerase 2 is going to work its way along the coding strand and produce a piece of mRNA, okay, which is complementary to that coding strand. So here is that piece of mRNA, okay, in red. And this piece of mRNA is now going to be used to make the protein. However, it is not currently ready for translation to occur. Okay, you can't just put this piece of RNA through a ribosome and expect the results to be good. Okay, this is what's known as a pre-mRNA, or the primary transcript of RNA. Okay, and the reason you can't just put this straight through a ribosome and translate it into a sequence of amino acids is that there are all sorts of portions within this pre-mRNA that are not supposed to actually be translated. Okay, so there are portions that are supposed to be translated, and I'll box these portions. So let's say that these three boxes here represent the portions that are supposed to be translated, and these will be the exons. So the portions that are actually supposed to be used in translation are called the exons, and then the portions that are in between the exons and are not supposed to actually be translated, these are called the introns. Okay, so what now needs to happen is you need to do some post-transcriptional modifications. You need to cut out the introns of your on your piece of pre-mRNA, and you need to glue the exons back together afterwards, and then you'll get a much smaller piece of mRNA, shown here, known as a mature mRNA. Okay, so this is a piece of mature mRNA. And this process of cutting out the introns and then gluing the exons back together, this is known as splicing. Now, we want to discuss the concept of having different splice variants. Okay, so the concept of having different splice variants is that there are, in some cases, okay, such as with the Reb gene, there is more than one way that you can splice the piece of pre-mRNA. Okay, so when you are confronted with this piece of pre-mRNA and you're going to make a piece of mature mRNA, there is more than one way you can actually cut it up and produce a piece of mature mRNA. And this will result in, these different splice patterns will result in different pieces of mature mRNA being formed. Okay, so to give you a very easy to understand example of this, okay, let's imagine that one of these exons, we'll say it's this middle one here, is not essential to be included. Okay, so you don't need to include this exon. Instead, you can cut it out as though it was an intron, basically. So what you could do is you could just join this first exon here to the second exon and get a smaller piece of mature mRNA. Okay, so here's a smaller piece of mature mRNA, and then this piece of mature mRNA could then be translated just like this initial piece here. Okay, and depending on which mature mRNA you produce from your pre-mRNA, okay, you will get a slightly different protein. So these will produce different proteins, and this is the way that you get different splice variants from the same gene. So one gene can produce more than one type of protein. This is what has happened in the case of Reb, okay? So these Reb alpha, Reb beta, and Reb delta, they're all from the same gene, they're just different splice variants. And indeed, Reb delta literally is what I've described to you here. It is the case that you have deleted a single exon from the piece of pre-mRNA, okay? And you therefore end up with a slightly shorter piece of mature mRNA and a slightly shorter protein than for Reb alpha. Reb alpha is the one where you include absolutely every single exon, whereas Reb delta you delete one. Reb beta is a little bit more complicated, you delete quite a few in Reb beta. Okay, right. So there are these different forms of Reb. Now they all pretty much do the same thing as far as we're concerned. These are found all over the body. They're pretty much evenly distributed everywhere. Okay? Whereas the final forms of Reb, and I want to stress there are other splice variants other than the alpha, beta, and delta Reb. Okay? Those are much more niche. They're mainly found in the testes, and they don't seem to do the same thing as these three. So these three all seem to do the same thing, so we're going to concentrate on these three. Okay, right. So we now want to talk about the structure of a Reb protein. Now, of course, all three of these are going to have slightly different structures. Reb alpha is the one that contains everything, so we're going to start with Reb alpha basically, and then I'll tell you where Reb delta has a single piece missing. They will all have the basic essentials that they need to perform their role. Okay, so now let's have a look at the structure then of our Reb protein. So we'll start with the amino terminus of the Reb protein here, and we're drawing at the moment Reb alpha, remember, the biggest splice variant. So this is the one where you include all the exons. And I should just add there are actually 11 exons in the case of the Reb gene. I didn't draw that, but you would have 11 of these boxes rather than just 3. Okay, right. So here's the amino terminus of our Reb alpha protein. Following the amino terminus, you have a region known as Q1, okay? And Q1 is known as the glutamine-rich region 1. So Q is the single-letter amino acid code for the amino acid glutamine, okay? So this is the glutamine-rich region 1, okay? Also called Q1. So let me just show you the structure of a glutamine amino acid residue. Okay, so here is the core amino acid structure. Here's the amino group, the alpha carbon with a hydrogen coming off it, the carboxylic acid group, and then the R group of glutamine consists of 2 methylene groups, okay, like so, and then a primary amide group off the side of that, so a carboxylic acid group with an amino group coming off. So it's like glutamate, in fact, but instead of having the carboxylic acid group here, instead the carboxylic acid group has been reacted with an ammonia molecule to create glutamine here, the primary amide. Okay, and the single letter code for glutamine is Q, which is why this region is called Q1. Okay, so the significance of Q1 isn't totally understood, okay? So it's kind of just a filler region, but it does have these glutamines in. Okay, so at the moment we understand it just as a region, basically, which has a lot of glutamines in, but it's not one of the ones that is essential for the function. Okay, then you have the region that is deleted in Krebs delta, so this certainly isn't essential for Krebs alpha function. Okay, and this is known as the alpha peptide here. Okay, so remember, Krebs alpha contains all 11 exons, it's the full protein. In Krebs delta, you've deleted one of the exons, and that deletion of the exon results in the removal of this alpha peptide. Okay, right, and this alpha peptide is 14 amino acid residues long. So the deletion of one exon from the pre-mRNA to produce the mature mRNA that will code for a Krebs delta removes 14 amino acids from the Krebs delta, but otherwise it's essential to Krebs alpha. It just identical, to Krebs alpha. It just doesn't have this alpha peptide. Okay, after the alpha peptide, we then have an important region, okay, known as the KID. Okay, and this is the portion that's going to be modified by calcium calmodulin dependent kinase. Okay, so this is called the kinase inducible domain. Okay, so K stands for kinase, I stands for inducible, and then D stands for domain. So for short, KID. Okay, and this is going to contain an important serine residue, so I'll put this here, known as serine 133. Okay, so this is serine 133, and serine 133 is the one that's going to be phosphorylated by the calcium calmodulin dependent kinase to activate the Krebs protein. Okay, but we'll come back to that later. After the KID domain, what you then have is another glutamine-rich region known as Q2. Okay, so I'll colour in Q2 in yellow here. So again, this just has a lot of glutamines in, which is why it's called a glutamine-rich region. And it's the second one, so it's called the glutamine-rich region 2. Now, the next portion is really important. Okay, so the next portion we'll divide into 2 separate portions, and then you finish with the carboxylic acid terminus here. So we're going to start off with this final portion here. Now, Krebs proteins have to dimerize in order to be functional, okay? And this domain down here is going to be incredibly important for the dimerization of Krebs proteins. Okay, so this portion is what's known as a leucine zipper, okay? And this basically is an alpha helical structure. In fact, the whole protein folds into an alpha helix. So this portion in particular is a part of this great alpha helix. Okay, and basically the reason it's called a leucine zipper is that it contains leucine residues at every 7th position. Okay, so let me just show you what the structure of a leucine residue is. So here's the amino group of this amino acid. So we'll start again with the core amino acid structure. Then we have the alpha carbon with a hydrogen coming off it, the carboxylic acid group down here, and then the R group of a leucine amino acid residue consists of a methylene group, then with another carbon coming off, which will have 2 methyl groups attached to it, 1, 2, and then a hydrogen also coming off it. So the R group of leucine residues is extremely hydrophobic. Okay, so the idea is that within this leucine zipper region of the Krebs protein, what you will have is leucine residues every 7th amino acid. Okay, so let's say this amino acid here is a leucine, then you'll count up another 7 amino acids. You'll go 1, 2, 3, 4, 5, 6, 7, and then you'll have another leucine residue. Now, I've over-exaggerated here. There will be more amino acids than 7 in between there, but sorry, there will be less amino acids than 7 in between there, but the point is that after 7 amino acids, you then have another leucine residue. So this is a leucine residue in green, this is a leucine residue in green. You then count another 7 residues along, and you'll get another leucine residue. So every 7 amino acids, you'll have a leucine. So if this is 1, 2, 3, 4, 5, 6, 7, 8, then if this one is a leucine, you'll then go 1, 2, 3, 4, 5, 6, and then the 7th one will be a leucine. Okay, so you have leucines separated apart by these 6 amino acids in between them, and that's a leucine residue every 7th amino acid. Okay, so in this leucine zipper region then, you have leucine residues every 7th amino acid, and this is going to be really important in dimerizing Reb proteins together. Okay, so all 3 of our Reb proteins, Reb alpha, Reb beta, Reb delta, they all have these leucine zipper regions. Okay, and what's going to happen is at the leucine zipper region, you're going to get the 2 Reb proteins binding together. Okay, and the way this is thought to happen is that both of these leucine zipper portions here in green are alpha helices, and they've got leucine residues sticking out the side of them. Okay, and the idea is that you'll bring another one of these over here. Okay, which will also have leucine residues sticking off the side of it. And the idea is that the leucine residues will interact with each other very favourably, basically, because they're both hydrophobic structures, and that will hold the 2 leucine zipper alpha helices together. Okay, so that's this portion of the Reb protein then, and that's present in all Reb proteins. You always have this leucine zipper, which is responsible for dimerizing 2 Reb proteins together. Okay, right. Then, this domain between Q2 and the leucine zipper here is called the basic DNA binding region, and we'll continue this discussion in the next video.